What is the role of photosynthetic pigments? To absorb electromagnetic radiation and transmit the energy to the reaction center in photosystems. 2. Why do plants appear green? Chlorophyll does not absorb green wavelengths, instead, it reflects green light. 3. What instrument measures the light absorption ability of pigments? Spectrophotometer. 4. What is the primary role of chlorophyll-a? It absorbs and directly converts light energy into chemical energy in the form of ATP and NADPH. 5. What are accessory pigments? Pigments like chlorophyll-b, xanthophylls, and carotenes that absorb light energy but cannot convert it into chemical energy. 6. What is the function of accessory pigments? They transfer absorbed light energy to chlorophyll and assist in producing more chemical energy. 7. How do some carotenoids protect chlorophyll? By absorbing and dispersing excessive light energy. Absorption spectrum. 8. What is an absorption spectrum? A graph showing the relative absorption of different wavelengths of light by a pigment. 9. Which wavelengths do chlorophyll-a and chlorophyll-b absorb most? Violet blue, 400-470 nanometers, and orange red, 630-660 nanometers. 10. Which wavelengths do carotenoids absorb most? Between 430 nanometers and 500 nanometers. 11. What causes the difference in absorption spectra of chlorophyll-a and chlorophyll-b? Differences in molecular formulae, structural arrangements, and physical properties. Arrangement of pigments, photosystems. 12. Where are photosynthetic pigments located? On the surface of thylakoid membranes. 13. What are photosystems? Clusters of pigment molecules embedded in thylakoid membranes for efficient absorption of light. 14. What is the antenna complex? The peripheral part of a photosystem containing accessory pigments. 15. What is the reaction center? The central part of a photosystem containing chlorophyll and associated proteins. 16. What is the optimal absorption wavelength of PS1 and PS2? PS1, 700 nanometers, P70, PS2, 680 nanometers, P680. 17. Why is the order of photosystems named PS1 and PS2? Based on the order of their discovery, not their arrangement in the thylakoid membrane. 18. How do pigments in the antenna complex assist chlorophyll-a? They absorb light of various wavelengths and transfer energy to chlorophyll-a. 19. What happens when energy excites chlorophyll-a electrons? The excited electrons escape and are accepted by the first molecule of an electron transport chain. Overall mechanism of photosynthesis. 20. What are the two phases of photosynthesis? Light-dependent phase, light reaction, and light-independent phase, dark reaction. 21. Where does the light-dependent phase occur? In the thylakoid membranes. 22. What are the inputs and outputs of the light-dependent phase? Inputs: light and water. Outputs: ATP, NADPH, and oxygen. 23. What is the input and output of the light-independent phase? Input: CO2. Output: glyceraldehyde 3-phosphate. 24. Why is the dark reaction called light-independent? It does not directly require light but depends on ATP and NADPH from the light reaction. Light-dependent phase. 25. What are the two types of electron flow in the light reaction? Non-cyclic and cyclic electron flow. 26. What is photophosphorylation? The production of ATP during the light reaction. 27. What is non-cyclic photophosphorylation? A process where electrons flow from PS2 to PS1, producing ATP and NADPH. 28. What is cyclic photophosphorylation? A process where electrons flow back to PS1, producing only ATP. 29. What is photolysis of water? The splitting of water into protons, electrons, and oxygen in the presence of sunlight. 30. What is the Z scheme? A conceptual zigzag path of electron transport during non-cyclic photophosphorylation. Light-independent phase, dark reaction. 31. What is the role of NADPH and ATP in the dark reaction? NADPH reduces CO2, and ATP provides energy for carbon fixation. 32. What is the Calvin cycle? A metabolic pathway where CO2 is reduced to form glyceraldehyde 3-phosphate. 33. What are the three phases of the Calvin cycle? Carbon fixation, reduction, and regeneration of CO2 acceptor. 34. What is the first stable compound of the Calvin cycle? 3-phosphoglycerate, 3-PGA. 35. What is ribulose bisphosphate, RUBP? A 5-carbon sugar that acts as a CO2 acceptor. 36. Which enzyme catalyzes carbon fixation? Rubisco, ribulose bisphosphate carboxylase, oxygenase. 37. What is the final product of the Calvin cycle? Glyceraldehyde 3-phosphate. 38. How many molecules of G3P are needed to form one glucose molecule? 2 molecules. 39. What is the overall equation of the Calvin cycle? 3CO2 plus 6 NADPH plus 9 ATP, G3P plus 3H2O plus 9 ADP plus 9 pi plus 6 NADP. Thanks for watching.